We are starting the topic of respiration. So, respiration is a process where there is oxidation of certain organic compounds to get energy. Now, this energy can either be adenosine triphosphate ATP or it can be GTP. And there is inevitably some loss of energy in the form of heat. These ATP and GTP will be utilized in metabolism and life processes. These organic compounds in case of respiration are what we call respiratory substrate, and respiratory substrate includes carbohydrates, lipids, proteins and sometimes organic acids. Carbohydrates as well as lipids are the major respiratory substrates. Let's consider plant cell. Plant cell will form their own respiratory substrate. Light energy will help in the formation of this respiratory substrate. The process we call photosynthesis. The oxidation of respiratory substrate will give the plant energy in the form of ATP or GTP, and this energy will be utilized in the life processes, in the metabolic processes and storage as well, and that storage will be acting as the respiratory substrate for the animal cell, because the animals are taking this respiratory substrate, these organic compounds, as diet. Let's talk about oxidation of respiratory substrate. So either there is partial oxidation or there is complete oxidation. In case of complete oxidation, there is liberation of carbon dioxide and all the rest of carbons are being oxidized and we get energy. But in case of partial oxidation, there is formation of intermediate products, and these intermediate products, they may be alcohols, they may be aldehyde, they may be acids. Hydrocarbons are our respiratory substrates. Hydrocarbons will oxidize to form alcohols. If alcohols oxidize, they will form aldehydes. If aldehydes oxidize, they will form acids, and these acids will be oxidizing to form carbon dioxide as final product. And this is the process of complete oxidation. What if we take it in reverse? Carbon dioxide will reduce to form acids. Acids will reduce to form aldehydes, which will reduce to form alcohols, and finally alcohols will reduce from hydrocarbons. If the process is complete from hydrocarbons to carbon dioxide, we call it complete oxidation. And if it is not until carbon dioxide is produced, either it may be till the acids, it may be till aldehydes, it may be up to alcohols. In these cases, we call it incomplete oxidation. If the process is oxidation, there may be certain dependency on oxygen. So it can be oxygen dependent, it can be oxygen independent. If the process of respiration is oxygen dependent, we call it aerobic respiration. If it is oxygen independent, it can be either anaerobic respiration or it can be fermentation. Aerobic respiration will lead to complete oxidation. This aerobic respiration is present in both prokaryotes as well as eukaryotes. Anaerobic respiration is exclusive to prokaryotes. There are few exceptions in case of eukaryotes, like red blood cells in humans, human erythrocytes, which lack nucleus, and there is no anaerobic respiration because they lack the mitochondrion as well, and there is no cytosolic mechanism as such which support the anaerobic respiration. But majority of the times it is only present in case of prokaryotes, and fermentation can be seen in both prokaryotes as well as eukaryotes. Let's talk about the equation of complete oxidation. The respiratory substrate, I'm taking glucose in this case. The process has to be aerobic respiration. In order to be complete oxidation, we will get carbon dioxide and we will get energy. Again, energy will be in the two forms, one which is ATP or GTP. There will be loss as heat. Now, how much ATP will be produced? It also depends. There may be production of either 32 ATPs or there may be production of 30 ATPs. So this is the equation of complete oxidation, or this is the equation of aerobic respiration. Let's talk about the site of respiratory processes with respect to prokaryotes as well as eukaryotes. So there are three fundamental processes. The first one is glycolysis. Second one is citric acid cycle, which is also called Krebs cycle. And last one is oxidative phosphorylation, which is sometimes referred as the electron transport system. In both prokaryotes as well as eukaryotes, the glycolysis will take place in the cytosol, whereas in case of citric acid cycle, eukaryotes have mitochondrion. Prokaryotes lack mitochondrion. Citric acid cycle will take place in cytosol as well. If we talk about oxidative phosphorylation, there are certain enzymes involved which are membrane bound enzymes. In case of eukaryotes, they are in inner membrane of mitochondrion, Whereas in case of prokaryotes, these enzymes are present, or these enzymes are embedded in the plasma membrane itself. Let's talk about the general features of some metabolic pathways. Metabolic pathway can be reversible or it can be irreversible. In case of reversible pathway, the single enzyme, which is E1 in this case, is carrying the reaction forward. Reactant A is converting into product B, and the same enzyme E1 will also be able to carry this pathway in the backward direction, so that the reactant B can be converted to product A. Another thing which has to be considered is, in case of reversible pathway, each and every step of the metabolic pathway has to be reversible. If there are 10 steps in a metabolic pathway, each and every step of that pathway has to be reversible. In case of irreversible pathway, let's take the same example, the product A and B. Either this process is thermodynamically favorable in the forward direction only, which means that A can only be converted to B. Let's take the example of enzyme E1 as well, and backward reaction is not feasible thermodynamically. Or there is another case in which backward reaction is possible, like A is converting into B and B is reverting back to A, but the main difference is the enzyme which is converting the reactant A into product B, E1, is not responsible for carrying the reaction backward, which means that we need another enzyme. For example, enzyme E2 is converting the reactant B into product A, which is the backward side of the reaction. This case is also called irreversible. And another point to remember, if a metabolic pathway is having 10 steps, for example, and if only one step is irreversible, the whole metabolic pathway will be considered irreversible. Let's talk about regulation of enzyme catalysis. We must understand the basics of enzyme regulation in context of the metabolism. The detailed topic we will discuss in the enzyme itself. The regulation of enzyme catalysis can take place at two different levels. Either we regulate the metabolic pathway through regulating enzymes at the level of their synthesis, or we regulate the pathway by regulating enzymes at the level of their catalysis. Each enzyme has to have certain specific gene to be synthesized. For example, in this case, enzyme A is being synthesized by the gene A. We can regulate this process at the level of transcription or translation. We can either inhibit this process, where the basal transcription rate is repressed, or we can upregulate this process where the transcription rate is enhanced. And this can be done at both the levels, either at transcription or translation or both. Some enzymes only work at the level of synthesis, and some enzymes are being regulated at the level of enzyme catalysis, by which we mean that enzyme is constitutively synthesized all the time. Enzyme is made all the time. The gene is always on, there is always basal level of transcription, but we are regulating the activity of this enzyme, and this can be done by two processes, either by the covalent modification. By covalent modification, we mean that we will be attaching some kind of molecule through covalent bond, like we are attaching phosphate here, and this attachment of phosphate may either upregulate the pathway, which means the process of catalysis is increasing, or it may downregulate the pathway, which means the process of catalysis is being decreased. And second way is non-covalent modification. Now, it has to be noted that both these covalent as well as non-covalent modifications have to be reversible in nature, and this non-covalent modification is also termed as allosteric regulation. And allosteric regulation works in a way that at some specific allosteric site there will be binding of some group, and this binding will be non-covalent in nature and reversible in nature. The enzymes which are regulated by this allosteric regulation are non-Michaelis-Menten enzymes. These enzymes will not follow the Michaelis-Menten kinetics. Let's talk about some common names of enzymes which are involved in oxidation reduction reactions. These enzymes in which the substrate is getting oxidized and the electron acceptor is oxygen itself, we call such enzymes oxidases. If there is physical addition of oxygen either as an atom or molecule, we call such enzymes oxygenases. If the electron acceptor is hydrogen peroxide, we call such enzymes peroxidases. And if the electron acceptor is like some molecules like NAD, FAD or lipoate, we call such enzymes dehydrogenases. Let's talk about another terminology regarding phosphorylation. There is substrate level phosphorylation, there is oxidative phosphorylation, and there is photophosphorylation. In case of substrate phosphorylation, it is the substrate which is donating the phosphate. For example, the substrate is donating phosphate to ADP and this ADP is converting into ATP. So we can get energy. This is what we call substrate level phosphorylation. Also, it has to be noted that the phosphate which is being donated by the substrate, this phosphate is organic phosphate. What happens in case of oxidative phosphorylation? We have similar equation. A is oxidized into B, and by the process of oxidation there is a release of some energy, and this energy is being utilized in the phosphorylation of ADP. So we get ATP. But the main difference from the substrate level phosphorylation is that the phosphate which is being utilized is inorganic phosphate. If we see this process in a bit more detail, so there is oxidation. For example, NADH is being oxidized into NAD. There is loss of electron. This electron is being accepted by oxygen. There is formation of water, and due to these energy differences there is creation of electrochemical proton gradient which will be helping in protomotive force, and this protomotive force will be responsible in attaching phosphate to ADP, and there is formation of ATP. But this phosphate is inorganic phosphate, whereas in case of substrate level phosphorylation, the phosphate is organic phosphate because it is attached to this organic molecule. Here light energy is harvested by the chlorophyll. There is involvement of some kind of electron transport system, and there is formation of again electrochemical proton gradient. There will be protomotive force, and due to this protomotive force there is the pumping in of protons and attachment of phosphate which is inorganic phosphate to ADP, and there will be formation of ATP. So the difference between oxidative and photophosphorylation is that the energy which is utilized is coming from the process of oxidation in case of oxidative phosphorylation, and in case of photophosphorylation the energy is harvested from the light energy, from the light source, and in both these cases there is addition of inorganic phosphate which is present in the cytosol itself. Whereas in case of substrate level phosphorylation, the phosphate is directly donated by the substrate itself. This is the high energy phosphate which is directly attached to the substrate itself, and this phosphate will be attached to ADP, and there will be formation of ATP. So these are the three basic types of phosphorylations we study in case of metabolism.